Okay, so welcome back from the break. Hopefully you went and got a drink or got up and stretched at least. And you're now ready to continue. So let's take a look at the concept check questions and see what the answers to these are. So why are plasmids valuable tools for creating recombinant DNA? Because you can cut them with restriction enzymes and you can cut the DNA that you want to introduce with the same restriction enzymes to produce sticky ends that allows you to easily insert the foreign DNA into the plasmid. But then remember, of course, the plasmids then are easily taken up by bacteria from the environment. And you can increase the likelihood of that by using that heat shock technique that I talked about that increases the likelihood of taking up the plasmid. So you can engineer the plasmid very easily using restriction enzymes to introduce the genes of interest, and then you can very quickly and easily get them incorporated into bacteria by using what is known as a transformation technique. The uptake of that foreign DNA is actually referred to as transformation because it transforms the cell by introducing a new gene sequence. It changes the phenotype of the cell. So restriction enzyme cuts DNA at the sequence AATC. How many cuts would this enzyme make in that sequence? Well, all we need to do is identify where that sequence occurs, and it occurs twice in this sequence. So there's number 1, and there's number 2. So it would cut twice. Bacteria are easier to manipulate than yeast. So why are yeast used to produce some human proteins? So here you need to know something about yeast that I haven't told you. You remember we talked about plasmids for engineering bacteria. Well, there is a yeast equivalent to bacterial plasmids. So it's called a YAC, which stands for yeast auxiliary chromosome. It's kind of the yeast equivalent of a plasmid, and it can be genetically engineered as well. So why go to the bother of engineering a yeast, which is not as easy to do? It's a lot harder to get YACs into yeast than it is to get plasmids into bacteria. Why go to all the trouble of introducing a gene via a YAC into yeasts? Well, if the gene of interest is a eukaryotic gene, remember we have to take into account that eukaryotic genes include introns and exons. And part of the processing of a transcript of a eukaryotic gene is to remove the introns and splice the exons together. That you don't have to do in a bacterium because a bacterial gene does not contain introns. It's all coding. So if you want to put a gene into a bacterium that comes out of a eukaryotic organism, like the human insulin gene, you can't just cut out the human gene from the chromosome and plop that into a plasmid and expect the bacterium to make sense of it, because it doesn't recognize introns. Introns have no meaning to bacteria. And so you actually have to engineer a human insulin gene, taking out the introns and splicing the exons together. You actually have to engineer the DNA sequence, cut out the nucleotides that represent the introns, and then splice together the nucleotides that represent the exons. In other words, you have to make a synthetic DNA sequence that represents the insulin gene minus its introns before you can insert it into the bacterium. A yeast, on the other hand, well, that's a fungus. Yeah, yeast is a fungus. It's a relative of mushrooms. And it's a eukaryote. So it understands introns. And it has the system for taking out the introns and splicing the exons together. So if you can get the gene into a yeast, you don't have to worry about cutting out introns and splicing exons. The yeast will do that for you. And so it is sometimes really worthwhile persevering with getting the gene into a yak and then getting the yak into a yeast, simply because you then let the yeast do the work of assembling the intact protein by editing the transcript to create the messenger RNA by cutting out the introns and splicing the exons together. And if it happens to be a human gene where there are lots of introns and lots of exons to be spliced together, it actually takes less work to put it through a yak and into a yeast than it would take to edit the DNA, because editing DNA up until relatively recently was a little bit of a chore. So that's the reason why you would persevere and put it into a yeast rather than put it into a bacterium, even though the bacteria are a lot easier to manipulate. So what about genetic engineering in agriculture? Because this is one that a lot of people talk about. Genetically modified plants, not so much genetically modified animals, because it's a little bit harder to genetically modified animals, and until recently, the USDA had not approved any genetically modified animals for sale in the United States. However, that has changed just recently. A genetically modified salmon is now being farm raised for sale in the United States, and a gene has been inserted into that salmon that means that it matures much more rapidly. In other words, it reaches saleable size much faster. Instead of taking 3 years to reach saleable size, it takes about a year to a year and a half. That means you don't have to spend as long growing the animals in large pens in the ocean in order to get salmon that you can then harvest and prepare for sale in supermarkets. So a lot of some farm-raised salmon these days is genetically modified, but you won't know which it is because there is no requirement so far from either the USDA or the FDA or any other regulatory body that requires the labeling of food as being GMO, genetically modified. Although a lot of supermarkets will tell you that stuff is not GMO, but they are not required to indicate that it is GMO. Nor should you assume that just because it's not labeled as non-GMO that it actually is. That being said, however, if you are eating products that contain anything from corn, soybean, cotton, canola, these are all today almost all genetically modified. So if you like your corn chips, if you use anything that contains corn oil or soybean proteins, cottonseed oil, canola oil, or anything that's derived from those, then you are almost certainly consuming products from a genetically modified organism. So here is a corn plant that has been attacked by a corn borer beetle. This is the larval stage of it. These corn have actually been modified to produce their own insecticides. This is something that a number of agricultural plants have now been modified to do. And you might ask, insecticides? What do you mean, insecticides? Well, if you have ever used a can of insect spray that contains chemicals called pyrethrins. Then you've been spraying an insecticide that was originally derived from daisies, because daisies are the original source of pyrethrins. Daisies make these chemicals as a natural insecticide. We have simply figured out how to produce them synthetically, and they were incorporated into cans of spray insecticide. But because the pyrethrins were produced as a consequence of genes in the daisies, once those genes were identified, it was relatively easy then to transfer those genes into other agriculturally important organisms, and those plants then started making their own pyrethrins that could protect them against insects. They started making their own natural insecticides, and pyrethrins are natural insecticides. Just not every plant makes them. Daisies are the ones that do, but if you genetically modify agricultural plants with the daisies' natural insecticides, they become insecticide producers themselves. Then we know strawberries are very, very susceptible to cold weather, to hard freezes. And unfortunately, strawberries produce their berries right at the end of winter, early in spring, so they can be very easily subject to hard freezes. And even here in Florida, where we have our strawberries growing over in the Tampa area, they can be subject to hard freezes and loss of the strawberries. But the bacterium that I referred to earlier, the Bacillus thuringiensis, produces a natural antifreeze. And the gene for that chemical has been identified, and strawberries have been genetically modified with this gene so that they now make their own natural antifreeze that stops them from being frozen if they get subjected to a hard freeze, a frost. So a frost is when you get, you've seen a frost, you get the frozen water on the surface of the plants. A hard freeze is when the temperature remains below freezing for several hours, and then the plant tissue actually starts to freeze. That's when the damage gets done. If it's just a light frost, the temperature doesn't drop below zero for an extended period of time, then a lot of plants will survive that. But a hard freeze, citrus plants, strawberries, lots of plants are susceptible to hard freezes. They can survive a light freeze, but they can't survive a hard freeze. So being able to produce your own natural antifreeze is a benefit. Potatoes and rice have been modified to provide a vaccine against cholera. So they have a surface protein that comes from the cholera bacterium. So this is a protein that's on the surface of the cells. When you ingest the protein, it triggers your immune system to make an antibody against it, so that should you be confronted with the cholera bacterium, your immune system already knows how to deal with it and kills it off, because cholera is really, really serious. It kills you very quickly. And the reason why is because the cholera bacterium affects the ability of your body to retain water. It actually triggers water to flow from the body into the digestive tract to try and counteract the effect of the cholera bacterium. That gives you diarrhea, and you become very, very quickly dehydrated, and you die of that dehydration as a consequence of the cholera. And cholera is endemic in several regions in Asia. So in Bangladesh, the Indus River Valley, in fact, so northern India and Bangladesh is where you find a lot of outbreaks of cholera. You also find it in Nepal, and then in a number of locations in South America and the Caribbean have unfortunately also developed different strains of cholera. And so by using potatoes and rice, the two most common forms of starch in people's diet and the source of a large number of calories in people's diet. And incorporating this cholera protein, the bacterial protein, into potatoes and rice means that people naturally, by eating just potatoes and rice, which they're going to do all the time, expose their immune system to this protein. The immune system then reacts against it, develops the ability to make antibodies against the cholera bacterium, so that when they get exposed to cholera, which they will on a regular basis because of contaminated water sources predominantly, no harm, no foul. The immune system knows how to deal with the cholera bacterium and kills it off before an individual becomes sick and dies of the effect of the dehydration. Then another example, and I'll show you a picture of these two, is what is referred to as golden rice. And you can see the golden rice. Here's normal rice, here's golden rice. You can see why it's called golden rice. And then sweet potatoes. Now, you know that sweet potatoes are a natural orange-yellow color. That's because they contain a substance called carotene. Now, carotenes, and particularly beta-carotene, are important because they are the source of vitamin A. It's a symmetrical molecule, and when you cut it in half, the two halves are vitamin A molecules. So long as you get sufficient beta-carotene in your diet, you'll get sufficient vitamin A in your diet. But the problem in particularly Southeast Asia and a couple of other locations around the world where sweet potatoes are a significant source of calories is that you don't get sufficient quantities of beta-carotene. If you eat a lot of rice, you get virtually no beta-carotene. Rice is mostly starch, remember. And so rice and a small amount of vegetables, but mostly rice, doesn't give you enough vitamin A. And the problem with that is vitamin A is central to normal healthy vision. And lack of vitamin A is one of the most common sources of loss of vision. And it's easily preventable by simply increasing the amount of beta-carotene in the diet and therefore increasing the amount of vitamin A that's present. And so the Rice Institute, which is located in the Philippines, many years ago developed an initial version of golden rice that was to be marketed in Southeast Asia, but it was not well received because it had an unusual taste. So they went back to the drawing boards and created what is now called Golden Rice 2. And this is actually well received because it tastes like the regular rice, but it has this high level of beta-carotene in it, which means that people can continue to enjoy their traditional diet of rice and vegetables with a little bit of meat, and they don't have to worry about the type of vegetables. Most of them are green leafy vegetables that have some beta-carotene in it, but they don't have a lot of yellow or orange vegetables in their diet, which is where you get the beta-carotene from. And so by eating golden rice, they can supplement the beta-carotene and therefore the vitamin A. The same is true with the sweet potatoes. A lot of sweet potatoes don't have the bright yellow color of the beta-carotene. They have a yellow color, but it's not because of beta-carotene. But by introducing beta-carotene into the sweet potatoes, again, you accomplish the same thing as you accomplish with the golden rice. You increase the intake of beta-carotene, and therefore you increase the amount of vitamin A in the diet, and you subsequently prevent the most common form of loss of vision, lack of vitamin A. It's a lot easier than trying to encourage people to take vitamin A supplements in the form of pills and so forth. If all you need to do is just keep eating your food and you get what you need, then obviously it makes sense to go that route. Then it is possible to genetically engineer animals. As I said, up till now, the permission to sell genetically engineered animals or animal-derived food in the United States has essentially not been possible, although with the salmon, the approval of the salmon, and this is the first one, now it looks like the door is opening for the possibility of introducing other genetically modified animal products. So why would scientists want to engineer the development of larger muscles in cattle and chickens? Well, it's the muscle that's actually the meat. When you eat meat, it's actually the muscle that you're eating. Up till now, the only way to develop those muscles was to inject growth hormones. And there has been a lot of issues with the use of growth hormones in developing larger muscles on animals. If you look at those large chicken breasts that you can get in the supermarkets, I don't know of any chicken that has a breast muscle that big. There's only one way that those breast muscles can be developed to that extent, and that's with the use of hormone supplements. But if you genetically engineer the animals, you may be able to engineer them to produce those large muscles without having to use the synthetic hormones. Now, I suspect that it's probably engineering in a gene for overproducing the hormones that naturally occur in the animals. I don't know for sure. I don't know enough about the technology because it's not an area of my specialty, but I suspect that that's probably somewhere along the lines that they're thinking. Here's another interesting one. Why would you want to introduce omega-3 fatty acids? Well, omega-3 fatty acids are unsaturated fats that are known to be heart protective. So there are omega-3 and omega-6 unsaturated fatty acids. And it's the ratio of omega-3 and omega-6, and the numbers actually refer to the position of the unsaturated bonds in the fatty acids. It is the ratio of omega-3 to omega-6 that's important. You want a lot of omega-3 and not so much omega-6. Omega-6 fatty acids have been implicated in inflammation, which is actually going to promote the very thing that you want the omega-3 fatty acids to prevent heart disease and to prevent stroke. They're heart healthy. A little roundworm called C. habitus or C. elegans or Caenorhabditis elegans actually contains a gene that can convert fatty acids into omega-3 fatty acids. And researchers have figured out a way to introduce the gene into pigs. Hasn't been approved for human consumption. So you don't have to worry about genetically engineered pork chops. Not yet anyway, because they haven't been approved. But it's proof of concept again. It would make the meat healthier to eat. And here's the salmon situation. So we've talked about this. So it means that the salmon are now ready for harvest about 1.5 years earlier than they otherwise would be. So it takes half the time to grow to the same size that would be marketable. So if you are consuming farm-raised salmon, it's as I said, it's potentially possible that you are consuming some of this genetically modified salmon. So what about the use of DNA technology for correcting human disorders? So this, we don't talk about genetic modification of humans. We don't talk about GMO humans. But the technology is basically built around the same technology that is utilized to create GMO organisms. So it's taking advantage of what we know about genetically modifying plants and animals, but using it to create a way of correcting a genetic disorder so that a person can go on and live a normal, healthy life. So here's the basic way in which it's carried out. You take in this case, an RNA. That is a copy of the gene that needs to be inserted, and you're going to use an RNA virus. The RNA viruses tend to be more specific in the cells that they're going to target. In this case, we want to target bone marrow cells, and there's a specific reason why we want to target those bone marrow cells. You can see that there's a concept check question here, a checkpoint question. I actually have this in my concept check questions at the break after this section, so that's coming up. So I'm not going to answer that question now, but there is a reason why we want to target the bone marrow cells. So what do the bone marrow cells actually do? Well, bone marrow cells are the source of your red blood cells, your white blood cells, and other components of your blood throughout the whole of your life. So the cells in the bone marrow are constantly dividing to produce these cells over the course of your life. Because you constantly have to replace your red blood cells, you constantly have to produce new white blood cells and other components like the platelets and so forth. These are all derived from the bone marrow. So the bone marrow cells are typically the ones that are used as the target cells. So I've told you the reason why we want to target the bone marrow cells. But I'm not going to tell you what it is yet. See if you can figure it out. So here we're talking about the gene therapy that I mentioned right back at the beginning, the one that was first worked out in golden retrievers, and the proof of concept treatment in golden retrievers was so good that it has been now trialed in humans. So it says 2009 they started working on this. That was the proof of concept in the golden retrievers that had the genetic mutation. Just recently, I believe it was around 2018, 2019. It was very, very recently that it was actually approved for use in humans, and several human volunteers have undergone the same treatment. It's basically injecting the cells of the retina with this viral vector that carries the normal gene and the corrective mechanism to correct the defect in the cells of the retina, because it's the cells of the retina that contain the mutation that needs to be corrected. So the virus is injected into the cells of the retina or injected into the retina, and they go ahead and infect the cells of the retina, carrying with them the machinery for correcting the genetic defect. And at this stage, while we know that this works spectacularly well in the golden retrievers, it restored normal vision to the dogs. They only treated one eye at a time, and it was remarkable how quickly the dog's eyes responded to the treatment. This is the reason why, having demonstrated proof of concept in the dogs, it was approved for human trialing. At the moment, as best I know from my reading, they're still waiting to find out just how effective this is going to be in humans. Apparently the response has not been anywhere near as quick and as spectacular as it was in dogs. Again, the dogs were the test subjects, but dogs are not humans and humans are not dogs. And while the technology works really well for the dogs, it may not be, unfortunately, as effective in humans. But there is a lot of hope that this will actually work out. I suspect that the reason why not much has been said is simply because we've had to deal with the pandemic, and everyone's been focusing on the pandemic for the last 18 months. But hopefully something will be released about this trial, at least at some point in the future, because if it works, this will be the very first successful human gene therapy trial, because previously when gene therapy was tried, and the first attempt at human gene therapy was, I want to say about 15, 20 years ago now, was for the treatment of diabetes. And a young man with severe diabetes was agreed to be the test subject. And unfortunately, something went wrong with the treatment and it wound up killing him. And at that point, a moratorium, as you might expect, was put on all human gene therapy. And it's been in place right up until this trial now with the loss of vision gene. This is the first human gene therapy that I'm aware of where permission was given to actually go ahead and treat this using the viral technology. Now it says here that, again, a couple of other diseases have been treated with gene therapy. This has not been using the viral system. This has been using a couple of different types of technologies for getting genes into the target cells. So the blood disorder involving the hemoglobin gene, that was actually sickle cell. This was a treatment targeted at sickle cell, and there was only 1 or 2 patients, I believe, was involved in the study. And then the degenerative disease of the nervous system, that was multiple sclerosis, I believe, was the condition that was being targeted there. And again, these 2 trials were very much not even clinical trials. These were just volunteers in a laboratory-based system. So these are not even as far along as the vision correction system is in terms of human treatment. So these are still waiting for permission to go into clinical trial. These have been basically 1 or 2 subjects in a laboratory situation that volunteered to allow themselves to be treated in a laboratory situation. This is not even a clinical trial in the traditional sense. Whereas this one for treating blindness, this is a clinical trial. This is not a laboratory situation. This is a clinical trial that has been approved by the FDA. So here is the concept check question. So we're taking a quick break here. I will break in case you want to go get a drink or take a stretch. I believe I may do that because I've been sitting here for a little while. Maybe I'll take a break too. So here's the concept check question. And I'll see you on the other side of the break with the answer to this question. Why are bone marrow cells considered a good target for human gene therapy? It is that question. Okay. See you on the other side of the break with the answer.